Hey everyone, welcome back to Window Seat Studies. I hope you all are doing well. So we started discussing about immunology, where we discussed about immune system, the types of immunity that we possess. So we talked about innate immunity, the immunity that we possess since our birth, as well as we talked about adaptive immunity, the immunity that develops over the time due to the exposure to different pathogens in our lifespan. So we had an introduction about both of those immune systems, and we also discussed about the cells which are a part of those immune system. So both of those immune responses, they have the cellular component as well as the humoral components. So we talked about hematopoiesis, the process of generation of blood by the hematopoietic stem cells. So hematopoietic stem cells are the ones which are going to get differentiated to give rise to those mature blood cells. The blood cells such as red blood cells, white blood cells, as well as platelets. So this whole process is known as hematopoiesis. We learned about this process in detail. Then we also started discussing about the cells of immune systems, which are generated from those hematopoietic stem cells, which belong to different lineages. So we talked about how the myeloid lineage cells, such as granulocytes, as well as the natural killer cells, both of them, they are a part of innate immunity, whereas the lymphocytes, such as B lymphocytes and T lymphocytes, which belong to the lymphoid lineage, are the part of the adaptive immune system. So we learned about all of those cells in detail. And then we started discussing about organs of immune system. So there are organs which are the part of immune system which are going to either initiate the immune response, or these are the sites where those cells that we discussed previously, they are going to get matured. They are originating from there, and they are getting matured there. So development of those cells is seen. So that is why those organs where the immunocompetent cells, either they are generated or matured, or they are initiating their immune responses, they are referred as lymphoid organs. So we talked about lymphoid organs, specifically the primary lymphoid organs, which are the part of our immune system. So lymphoid organs are the organs where the lymphocytes or the immunocompetent cells, such as the natural killer cells or the monocytes or the dendritic cells, all of them, they will be generated and matured. So such lymphoid organs are referred as primary lymphoid organs, whereas the secondary lymphoid organs are the ones where the immune response will be initiated. So we discussed about lymphoid organs and how those lymphoid organs are categorized into two types: the primary lymphoid organs and the secondary lymphoid organs. The primary lymphoid organs are the ones where the immune cells, they are going to originate and they are going to get matured. So the primary lymphoid organs are two: the bone marrow in adult mammals is going to give rise to the all of the immune cells, specifically the ones which are part of innate immunity as well as the adaptive immunity. But the T cells, the T lymphocytes, they will be matured in the thymus. That is why that is also a part of primary lymphoid organs. So the bone marrow is going to, in the bone marrow we have the hematopoietic stem cells. So if the each cell in the 50,000 cells of the bone marrow is the hematopoietic stem cells. So hematopoietic stem cells are quite rare, but they have enormous proliferative capacity. So the bone marrow is the tissue where those hematopoietic stem cells reside, and they can even slip into the circulation due to the sinusoids that are present in the bone marrow. So the bone marrow is the site for maturation of granulocytes, which belong to the myeloid lineage. It is also the maturation site for monocytes. They also belong to the myeloid lineage. This is also a site for maturation of dendritic cells, even the macrophages, and for B cells. Okay, The T cells, they will be originated in the bone marrow from the hematopoietic stem cells. So those T cell precursors, they travel from the bone marrow to the thymus with the help of the circulation. And in the thymus, those T cell precursors, basically those thymocytes, they will form the mature T cells, which will be expressing either the CD4 molecules or the CD8 molecules. So thymus is the site where the T cells are going to get matured, and they are going to express either CD4 positive. They could be either CD4 positive, means they could express CD4 molecules, or they could express CD8 molecules. So these are the primary lymphoid organs, whereas the secondary lymphoid organs are the ones, they are scattered all over the body, and they are going to take care of the initiation of the immune responses. So the cells which are originated in the primary lymphoid organs and which are now mature which are getting mature in those microenvironments, they will travel to the secondary lymphoid organs, and these organs, secondary lymphoid organs, will be the site where those cells, they will be exposed to the antigens for first time in the life. And even the antigens which are which could be blood-borne antigens, or they could be resident antigens. So all of those antigens will be brought up to the secondary lymphoid organs, so that there will be those cells, immune cells, they will be encountering those antigens. So the first encounter of the antigen with the lymphocytes will be seen in the secondary lymphoid organs. So here in the secondary lymphoid organs, the initiation of the immune response will be seen. And the secondary lymphoid organs are spleen, that we are going to discuss today in detail. Then there's MALT, mucosal associated lymphoid tissue, and then there is lymph nodes, which are a part of the lymphatic system. So lymphatic system overall, it is also important for initiation of immune responses, because it is the one which is going to carry those antigen presenting cells, lymphocytes, in all over the body within a minute. So spleen and the lymph nodes, both of them, they are highly organized secondary lymphoid tissue, whereas the MALT, the mucosal associated lymphoid tissue, it is less organized compared to spleen and lymph node. And again, the way of functioning, way of presenting antigen, that will differ. That is why those they are different. They are present over the different locations. Whereas malt consists of this is present in the respiratory tract. This mucosal tissue is also present in the gastrointestinal tract. So this is present in respiratory tract, gastrointestinal tract. Okay, so the pairs, patches, the tonsils, they are the part of this tissue, and they are going to be the sites where the antigen will be facing those lymphocytes. Okay, so in today's lecture, we are going to only discuss about spleen in detail. We'll talk about the histology of spleen, the functions of the spleen, and then we in other lecture will discuss about lymph nodes, the lymphatic system, as well as the MALT. Okay, so let's get started with the lecture. I hope this whole overview is clear to you all. Let's discuss about spleen in detail. So spleen is also known as graveyard of RBC, and I'll come to that later, why it is referred as graveyard of RBC. But spleen has multiple roles besides initiating immune response, or besides providing an environment for the immune cells to to encounter those antigens. So spleen, the other jobs of the spleen are to filter the blood, to remove the RBC, to recycle them. Okay, so that is why that is also referred as graveyard of RBC, because the damaged red blood cells, the damaged erythrocytes, or the erythrocytes which are now old and which needs to be replaced, they will be degraded over here in the spleen, and that is why it is also known as graveyard of RBC. Basically, we are recycling all those RBC, the old RBC, or we are discarding the damaged RBC at this site. So besides initiating immune responses, spleen has other roles. So when I mentioned, it is for the filtration of blood. So it filters blood. Okay, blood filtration is one of the roles besides the initiation of immune responses. Then it also participates in hematopoiesis. Okay, so that is why spleen is really very important organ for us. But removal of spleen does not really have major issues in for an individual. So spleen can be removed surgically, and still, and that individual can lead a healthy life. Okay, but that doesn't mean that there will be no consequences. So loss of spleen does result into some consequences, specifically in children's. So it results into a condition known as OPSI, the where the post splenectomy infections will be seen. So specifically in children, if the spleen is removed, the surgical removal of spleen is known as splenectomy. Okay, ectomy means removal. So splenectomy is the surgical removal of spleen. So that children that if it doesn't have spleen, so the spleen after the surgical removal, it is going to it is going to pose a serious threat or the fatal condition for that individual, because it is going to lead to overwhelming post splenectomy infection. So children who has undergone splenectomy, they are they can undergo this OPSI condition. This is actually a medical terminology for overwhelming post-splenectomy infection. Post-splenectomy condition, inflammation, sorry, infection. Okay, so the individuals whose surgical removal of spleen is done, as the spleen has crucial role in fighting the antigens, blood-borne antigens specifically. So the blood-borne bacteria, they can damage that individual, and that will lead to the some infections which are termed under this umbrella term OPSI. So I will come to this later. But let's first now discuss about the roles of the spleen. And for discussing the spleen, let's first know about this tissue. So this particular organ, this is a fist-sized organ, and it is very soft. Okay, so this is a soft fist-sized organ which is present below your diaphragm. So it is present below diaphragm. Okay, behind stomach, in upper left abdomen. So in your abdominal area, specifically the left abdominal area, the spleen is situated just below the diaphragm, behind the stomach, on the upper side of the left abdomen. Okay, so I hope you can imagine imagine the positioning. So let's assume that this is a person, okay, and this is the torso of the person, okay, and let's say that this is the diaphragm. So this is the right side of the person, this is the left side of the person. So the spleen will be present over the left side of that person, just below the diaphragm, behind the stomach. Okay, so this is how a very small organ, the fist-sized spleen, is situated inside our body. So this spleen, and not just spleen, all of the secondary lymphoid tissue, they are compartmentalized from the outer environment using a fibrous capsule. So fibrous capsule is going to surround this organ, and not just this organ, all of the secondary lymphoid tissue, and that is going to encapsulate the spleen. So this spleen is encapsulated by a fibrous capsule. Okay, and in this fibrous capsule, there will be the tissue which is going to do its job. So let's first learn about the histology of the spleen. So let's take a look at the spleen. How does it look like? So let's assume that I'm drawing here a cross section of the spleen. So spleen is a small organ. Okay, so let me just draw it. So I'm just simply drawing a circle, and I'm going to show you how the spleen looks like. In this cross section, so this is this cross section of spleen. So spleen has two microenvironments. Okay, one is known as red pulp and other is known as white pulp. Okay, so this is the spleen, cross section of spleen, and in this spleen there will be entering the splenic artery. Okay, so this is splenic artery, and there will be entering the vein. Okay, so vein is going to take all of the deoxygenated blood from the spleen to the heart. Okay, so that's the job of the vein. So this is the vein, splenic vein and the splenic artery in the cross section of the spleen. So the area surrounding this artery, this splenic artery, this particular area. Okay, let me just draw it in a simple way so that it will avoid the confusion. So let's draw this artery like this. Okay, and let's draw the vein like this. Okay, so the area surrounding this artery is known as PALS. So this is the area which is surrounding the artery, splenic artery. So I'm going to draw it like this. Okay, so this particular area surrounding the splenic artery is known as PALS, periarteriolar periarteriolar lymphoid sheath. So this lymphoid sheath is known as periarteriolar because it is, just beside, it is surrounding the splenic artery. Okay, so this is very important because this particular region is rich in the T cells. This is T cells rich region. Okay, so this area surrounding the PALS, so the area surrounding PALS, let me just have a different color for it. Okay, the area surrounding PALS, which I'm drawing in the orange color, okay, it is known as marginal zone. Okay, and there will be present follicles here and there near the marginal zone. Okay, so these are the blue ones, these are the primary follicles. Okay, so those three components, the PALS, the lymphoid sheath, the marginal zone, and primary follicles, they are a part of the white pulp. So they together make up the white pulp, and so this white pulp makes the 25% of the spleen, whereas the rest of the 75% of the spleen is the red pulp. Okay, so this remaining portion, this is the red pulp. Okay, so this is red in color because of the presence of sinusoids here. So red pulp has the sinusoids, which are the leaky blood vessels that allow passage of the cells through it. So, okay, so red pulp has sinusoids in it. Okay, sinusoids are the blood vessels which are leaky and which will allow the cells to pass through them. Okay, so because so that the cells, the macrophages, the dendritic cells, they will be present in the red pulp besides the blood, which is known as which is also known as peripheral blood. So sinusoids are the leaky blood vessels, so that they are going to leak the blood in that particular region, and which will release, so which will bring the all of the blood-borne antigen in the spleen, in the tissue of the spleen. So spleen is going to tackle the blood-borne antigens, means the antigens which are traveling in the circulation. Okay, the infections, the blood-borne infections, they will be tackled in the spleen. So they will be released in they will be released in this sinusoids, and they will be brought in by those arterioles as well. But no, the red pulp does not do the job of handling this infection, whereas red pulp has the job to remove the RBCs. Okay, so red pulp is not the part of the immune system. We can say that confirmly, because it is only participating in removing the old red blood cells and removing the damaged red blood cells. So here in the there will be there will be the cords of Bilirod, the splenic cords, which will form here the meshwork of the macrophages, dendritic cells. So in those macrophages and dendritic cells, they are going to degrade those RBC. They are going to degrade the hemoglobin, and that is why the iron containing pigments will be here, which will be giving it a red color. And again, there's blood present over here. So this red pulp basically has a part in blood filtration. So it is going to remove old damaged red blood cells, and it will also have macrophages and dendritic cells, which won't tackle the blood-borne antigens, but they will only engulf the old RBC, and they will degrade the hemoglobin to release the iron containing pigments. Okay, so whatever job the initiation of immune responses, that job will be done by the white pulp. So here in the white pulp, as I told you, the periarteriolar lymphoid sheath, it is the T cell rich zone. Okay, so the T cells, they will be in the ample amount in this PALS, and in the primary follicles, there will be the B cells. Okay, so primary follicles is the B cell rich region. Okay, so what happens is that whenever any antigen it is circulating in the blood, so that blood-borne infection, that antigen, that will come through the artery, the splenic artery is going to bring those blood-borne antigens alongside the lymphocytes. So lymphocytes plus those antigens plus the antigen presenting cells, all of them, they will be there in the blood, and they will enter in the spleen through this artery. Once they enter in this artery, the T cells that will slip in this PALS region, and that is why PALS region will be rich in the T cells. And here in this PALS region, those blood-borne antigens, they are going to face those naive T cells, and which were brought by the blood, and they are going to face those T cells which were naive previously with the help of MHC molecules, and this is how the T cells which are present in PALS region, they are going to get activated. Whereas those follicles, primary follicles, these are rich in the B cells, okay, which are there. So this is a microenvironment provided for B cells. So all of those B cells are naive B cells, means they have never undergone the antigenic interaction. But when this artery brings the antigen with it, so the antigen is going to enter into those primary follicles, and in this primary follicles, they will be trapped. Okay, so they will they will be trapped in marginal zone, and they will also be trapped in the primary follicles. So whatever antigens are being trapped in the marginal zone has some resident B cells. Okay, so those B cells are not brought by the circulation like the primary follicles or the PALS, but there are some resident B cells which are always there in the spleen, and they are present in the marginal zone. So these resident B cells in the marginal zones plus the macrophages which are present in the marginal zone, they are going to trap this blood-borne antigens, and they will fetch those blood-borne antigens to the primary follicles. Okay, what will they do? The antigens, some antigens will be tackled in this PALS region, some of them will be trapped by the marginal zone, and some of them will be trapped in the primary follicles. The antigens which are trapped in the marginal zone, they will be sent to these primary follicles, and once they are sent to the primary follicles where the B cells are rich, okay, so in this primary follicles, they are going to interact with those naive B cells, and the B cells will get activated, and they are going to secrete antibodies. So they will be trapped here, and they will be killed here. So these primary follicles, when they initiate the immune response, they get converted into what we call the structures. We refer them as the germinal centers. So these germinal centers are the structures which are B cell rich, and where the B cells are getting activated and they are trapping the antigens. So marginal zone is going to trap the blood-borne antigen. It is going to send them to the primary follicles so that the germinal centers will be formed in this by this primary follicles. Okay, so I hope the chronology is clear to you all. So the antigen and lymphocyte is going to enter through the artery, and not just so the T cells which are present here in the PALS region, they are also naive, means they are only expressing CD4 and they are expressing CD8 as well. So some cells are CD4 positive and some cells are CD8 positive. But those T cells only have those receptors and they have never undergone the antigenic interaction. Such T cells are known as naive T cells. So those naive T cells, they are going to face those antigens here and they are going to kill those antigens. And how the antigens will be encountered by the T cells, that encounter will be done with the help of MHC1 or MHC2 molecules. Okay, so we'll discuss about how the antigen presentation is done. We have discussed this in antigen presenting cells in detail, and we'll again be discussing about how that leads to the T cell activation in the T cell chapters. For now, what is important for you to understand is that whatever antigen or lymphocytes are entering in the spleen, that enters through the artery, and the T cells stay in the PALS region, B cells stay in the primary follicles, and the primary follicles, they will result into formation of germinal centers when the antigen is exposed to the B cells. Okay, so primary follicles, we can also call that they are rich in naive B cells, whereas the primary follicles which are going to form germinal centers in future, those germinal centers will be rich in the activated B cells, the B cells who has been exposed to the antigens and who are now synthesizing the antibodies, who are secreting the antibodies. Okay, so I hope this part is clear to you all. The marginal zone is rich in the resident B cells. So those B cells are not taken from the blood circulation, they are residing in the tissue. Okay, I hope that's clear. So marginal resident B cell as well as the macrophages, they are going to trap the antigens in marginal zone. So this is how the PALS along with the marginal zone and primary follicles, all of these three which are the part of phytopulp, they are going to initiate the immune responses. So here antigens will be presented to the cells, and the cells will get activated and they are going to kill the pathogens. So this is how the immunocompetent naive B cells and T cells, they get matured, they get antigen interaction and they get activated and they kill the pathogen, they tackle the blood-borne infection. So that is why for handling the systemic infection, means the blood-borne infections, spleen plays a very important role. So unlike any secondary lymphoid organs, so here I talked about the lymphoid organs, all of those such as malt and lymph node, they are associated with the lymphatic system. But spleen is the exception, the only secondary lymphoid organ which is not associated with the lymphatic system. So here when I discussed you about the cross section of the spleen, I did not mention any lymph duct entering or any lymphatic duct draining the lymph through the spleen. So neither the lymph enters nor the lymph is drained out from the spleen. So lymphatic system is not associated with the spleen. Only and only blood circulatory system is associated. So only as there is splenic artery and splenic vein which are going to let the blood in and they are going to take the blood out. Okay, so the only blood flow will be seen in the spleen, not the lymph flow. So lymphatic system is not associated with this secondary lymphoid organ, and still it is referred as lymphoid organ because the lymphoid organ does not mean the one where the immunocompetent cells, lymphocytes are going to get, they are going to activate the immune responses. But wherever immunocompetent cells are going to initiate response, they are known as lymphoid organs. So lymphoid organ does not necessarily mean that they are a part of lymphatic system or they are the ones where the lymphocytes are going to get activated. Whenever the immune cells, they are going to initiate the response in that organ, that particular organ is referred as lymphoid organs. That is why spleen is a best example of secondary lymphoid organ where the immune response will be initiated and still it is not associated with the lymphatic system. Rest of the secondary lymphoid organs, all of them, the Peyer's patches, tonsils, okay, the appendix, the lymph node, all of them, they are associated with the lymphatic system. And I'll talk about this how the lymph is, what is lymph actually, and how is the lymphatic system present in our body, and what are the lymph nodes, how this lymphatic system tackles the infection, and how the immune response will be initiated in the lymph node in the next lecture. And then we'll also talk about malt. So I'm done with this lecture here. So we have discussed about all bone marrow, we have discussed about thymus, and in this lecture we discussed about spleen. In next lecture we will discuss about rest of the secondary lymphoid organs. Till then, see you. Bye bye.